Well, now for something completely different. So I actually threw this together like 5 minutes before because I think I had heard, oh, you don't need a slide, and then, oh, yeah, maybe you need a few slides. So I may just stick on one slide for 10 minutes. But I want to talk a little bit about some of the work that my lab is doing at USDA, but more sort of in general, maybe agricultural concerns about climate change, how that may impact production on the high plains. That's where I do most of my work, but in other regions of the world, and why we're concerned or why we're not concerned about climate change. One of the interesting things that I go to a lot of ecology meetings. So the perspective when you talk about native ecosystems relative to certain issues with climate change and some of the issues we have in agriculture, it's much easier to work in agriculture and deal with some of these issues. So one thing that a lot of people miss when we talk about declining aquifer levels and climate change and how's that going to affect agriculture, we're around agriculture every day. If you live in New York, I lived in upstate New York, but was around people from New York City and in New York City for several years, or you live in a big city, most people don't think about where food comes from. I mean, Michelle Obama, I think, believes that the solution to a lot of our problems is urban gardening. They don't have any idea what type of effort, what type of inputs it takes to produce the amount of food, not necessarily required physically, but required economically to feed a nation of a couple 100 million people or other countries or whatever. So, but one of the things that you have to realize in terms of agriculture is it's a man-made endeavor. So our perspective on climate change, or at least my perspective on climate change, is much different than someone working in native ecosystems. And I've worked with John Zach and Dave Tissue in native ecosystems as well. But one of the things one of my colleagues, James Mahan, likes to say is agriculture is straight lines and right angles, right? So on the high plains, we need 100 days. We need 100 days to produce corn, sorghum, wheat, cotton. So do we care if that 100 days falls between, April and August, or if it falls between August and whenever? No, we don't really care about that. So climate change, in a simple sense, in terms of how season lengths are affected or how that impacts seasonal weather patterns and water use, again, it's a man-made endeavor. If we need to shift where we plant cotton, sorghum, and wheat, we'll do that. If we need to change the timing of that, we'll do that. So from that perspective, our dealing with some issues of climate change is much easier than what you have to deal with native ecosystems, because in a sense we have much more control over the environment as opposed to looking at how that's going to affect native systems. I'm going to go ahead and skip that since we had a whole talk on aquifers. But most of my comments will be directed towards groundwater use from the Ogallala Aquifer. So this region, we have it fluctuates, about half of our acres are irrigated here. This is just a stylized graph of yield and water, and this makes perfect sense. So as you increase water, yield goes up. Where this green hashed line is, that was our, I've got future condition on one side and the past or the current condition on the other side. The problem is we've been operating out here on this side of the graph for many years. So stable yields, we're working on part of a curve where as we add water, we get these increased yields, but we're working up on the plateau part of the curve where variation in water doesn't result in a huge variation in yield. Our current condition, though, is down on this steep part of the curve, right? So those are the issues we're dealing with now in terms of production. The good thing is any increase in water gives us a very sharp increase in yield. The problem is the margin for error is smaller. So our ability to manipulate cropping systems, to monitor stress in the field, to control that error is becoming more significant. And it's not just on the high plains, that's around the globe. So my lab works on we sort of straddle the fence. I'm trained as a biochemist and molecular biologist, but a significant portion of my work is in agronomy, which agronomy, I guess some part of my schooling has been in physiological plant ecology, and that's basically what agronomy is. So my lab looks at crop management approaches, how we can conserve water, how do we apply water, how do we monitor stress in the field, and then the other half of our efforts are on what genetics are available, what new biotechnology is available, and how can we merge these two approaches to improve production. So these are the types of questions that we ask. So this is from the plant perspective, very targeted approaches. So what do we think in terms of plant biology? What are the things that we want to look at to improve plant production on the high plains? The second question is, what type of diversity is out there? So one of the historical problems that's happened in plant breeding is you've had this very narrowing of germplasm. So we've bred over the last 1,000 years for yield. We want plants that grow big and make a lot of fruit or fiber. In doing that, what we've done is eliminated some of the genetic diversity for things like pest resistance, drought tolerance, pathogen resistance, and things like that. So we're going back and looking at wild species or older cultivated species and asking what type of genetic diversity is there. And then we work with the Genome Center here to look at genome-wide approaches to crop improvement. The real question is, and the reason that I'm involved with people like John Zach at the Climate Center and Lucia Barbado over here in GIS, is the questions we have, we want to work on a field scale or a system scale. And as a molecular biologist and a biochemist, that's not easy to do. We're used to working on a cellular and subcellular level. The problem is, if your problem is at a field scale, then your solution has to be at a field scale. And the biggest hurdle is this last part. How do we integrate or scale up to that systems level, whether it's a native ecosystem or a managed ecosystem, which is what a field is? What are the relevant production scenarios that we're dealing with? So we know how they've grown crops out here for the past 15 years. We want to know, how are you going to grow them in 5 years, 10 years, 15 years? What is the future of the Ogallala Aquifer for the next 20 years and the next 50 years? So that's why we're interested in predicted climate change scenarios, how accurately we can predict the life of the Ogallala in this region, how the Edwards Aquifer, so for places like the valley down areas like South Texas around Uvalde, where you have these high fluctuations, we need accurate models for predicting how groundwater resources change, how surface runoff is affecting groundwater resources and surface water resources. And using that to then select germplasm that's more adapted, or using biotechnology to develop germplasm that's more adapted to what we call future production scenarios. This is how much water you're going to have available. This is how these crops are going to have to be produced. You're only going to have X amount of water, and you need to apply them in this manner. So this is just one example of using sort of a field scale approach. So everybody that's flown into Lubbock during the winter knows that this is a center pivot. If you fly into Lubbock now, you see these beautiful dark green circles. A lot of times it looks like this. So if you look at, this is just to give you an example of what a crock goes through. This is a thermal image of that center pivot where we've planted peanut. And basically one of the things that we're interested in is this repeated cycle of optimal and suboptimal conditions and how that affects plant growth and how plants adapt to this cycle and recycle of stress. So this is plant canopy temperatures. So the blue area are plant canopy temperatures that are at an optimal temperature. So the pivot's moving in this direction. This is 1 day after an irrigation event. So 24 hours previous to this, the pivot was here, it moved here, and it irrigated this section of the field. So the plant canopies, the leaf temperatures, are sort of at an optimal temperature. As you move counterclockwise, the temperatures start to increase, and then these sort of concentric circles of different temperatures are where we've put down less water. And what you see is you get this pattern, this weekly pattern, and in some cases it's 2 weeks of this optimal conditions, moving into a drought stress, back to optimal conditions. So this, I just put this up here as an example of using sort of field scale remote sensing tools and then asking questions about how are the plants responding if we change this pattern or we change the amount of water we apply. How are the plants responding, and is there genetic diversity for how these plants respond? So this is meaningless, meaningless probably to most people here. But these are laboratory screening assays where we've looked for genetically diverse species of peanuts and asked, how do they respond to different stresses, thermal stress or drought stress? Do they respond differently? And would those work well in our stylized future production environments? And then this is sort of combining those two techniques. So now this is thermal images. These are plant canopy temperatures for season-long responses of different genotypes. And in this case, I'm showing one genotype under different water levels. So there are two axes here. There's a time of day axis going up here. So down here where it's blue, that's nighttime. Where it's red and green, that's plant canopy temperatures during the day. And then this is the entire season. So this would be day one of the season. This would be just prior to harvest. And I'm comparing 3 different years. So 2009 was sort of a wet year. 2011 out here was extremely dry and hot. 2010 was what I would say is sort of a normal year, sort of a moderately warm, but we had timely rains. So again, this is an application where we can monitor an entire field for an entire season, 24 hours a day, and work down at a plant level and ask, how are plants responding to the environment? Now, the other thing that we have at the top is air temperature. And this is work that we've done with James Mahan and Lucia Barbado here. And these are what we call time surface maps. So here we're looking at what does air temperature look like. We can measure lots of different environmental parameters, vapor pressure deficits, relative humidities. And we're going back and looking at seasonal variation. So how do seasons vary out here? In what's a decadal variation? What does the last 50 years look like? How far can we go back to get accurate environmental measurements, and ask what are the total number of environments that we potentially, our little 100 or 150 day environments, that we could potentially experience on the high plains. And how do we incorporate those, again, into growth models that allow us to integrate groundwater levels? Okay, this season looks like if you look at 50 years of seasons, how many seasons are actually different? Oh, there's only 5 different combinations of environmental perturbations that you see, and this looks like a season A. How does that affect plant growth? So I think I'm going to stop there. Surely I'm close to the end, but I always finish my talks with this slide, especially in ag meetings. So for us, in terms of the genetics, in terms of our ability to identify how plants respond, those tools are developed. Those tools are available. We have a really good handle on the biochemical and molecular responses to many of these stresses. On a field scale, again, that's our biggest hurdle. So our future in terms of production, in terms of tailoring germplasm for altered environments, it's all about identifying, quantifying, and managing stress. And to do that, it requires this, what we call a systems approach, integrating tools that, accurately predict water availability, accurately predict future climate, accurately predict, long-term weather forecasts. That's it.
Perfect. Thank you. Does anyone have any questions? Yeah.
So in the genetic modifications over hundreds of years, there's obviously genetically modified for advantage for the crop, but you're saying in the process of that it narrows the window, or that narrows the range of what different attributes they have, then that can have a negative effect that was not seen or not forecast?
Yeah, so in the simplest terms, historically what plant breeders have done is select for yield and done that under sort of optimal conditions. And what that unintentionally does is eliminates, again, some of the genes that would be associated with, for instance, drought tolerance. Now, again, if you think in terms, the other thing is, again, it's a man-made endeavor. So most of the plants that we're growing here are from subtropical or tropical origins, and they're perennial plants. We've taken them here, we've put them into a semi-arid environment. We grow them for 100 days as an annual. So we've manipulated, we've made agriculture fit our system. So we can certainly do that again. We just, in some cases, we need to take a step back and,
In some ways, we've bred show dogs, but they're not going to hold up in an alley fight.
That's true. Yeah, that's a good way to do it, to say it. Yeah. So the genetic diversity is there. It's just it's not there in commercial germplasm. So that's what I'm not a plant breeder, but that's basically what plant breeders are doing.
Yeah.
In looking forward and looking at options for the future, what sort of climate forecast models do you look at, or do you look at on the scale of a few years or decades or what? I
think right now, so I think a lot of people in here are going to know this, could answer that better than I can. Right now, I think the climatologists that we work with at USDA, I think is pretty comfortable with about 50 years of historical data in terms of the accuracy of that data. That's what we're comfortable with. I'm comfortable with it because he says that's what his model works best with that. Regionally, you have the nice thing is you have a huge amount of variation in a very small window, right? So we grow field trials from Brownfield, which is not far south of here. It's on the shallow end of the aquifer. So the slide I had previously said, the aquifer would be depleted in 50 years. Well, if you live in Terry County, it's 5, right? Most of those guys aren't going to be pumping water in 5 or 6 years. it can be 105 degrees there and they hadn't seen rain in 6 months. You drive to Plainview and it's 85 degrees. And so I think the modeling that we're doing now is really breaking this region up into sub-regions and asking how many possible environments do we see, and then making some extrapolation on, okay, this is the possible, these are the number of environments we could possibly see, this is how frequently they've occurred, and this is how they set up. The algorithms that run that I don't necessarily understand. But I think there's 50 years of fairly accurate climate data for this region. I don't know, John may know more about that than us, but.
So the vineyards in Terry County going to wither in 5 or 6 years?
I would say if they're putting significant amounts of water down on those vineyards, then that would be a serious concern. Now, again, most of those vineyards aren't nearly the size of a cotton or a peanut field, right? So if you've got, I work with growers that up around Plainview that say they'll run out of money before they run out of water. I work with guys in Terry County that have 10, 11 wells tied together, and they can run one pivot and make a circle in 14 days. It's just not a very, we're transitioning back to dry land production in those areas. Now, again, if you're growing 100 acres of grapes, you don't need the same amount of water. And it's a high-value crop. You could tie 9 wells together and put. So the other thing I think that a lot of people don't realize is, there's agronomy and there's biology and biochemistry and molecular biology, and there's agriculture. Agriculture is economics. So, I'll defend farmers on a certain level here is, hey, these guys waste water. These guys, why don't they run this with solar power? Why don't they run this with wind power? Trust me, farmers that are still in business here know every gadget, every computer program, every model that's available. When they take out a pen and a paper and, add this up, if there's a minus sign in front of the bottom value, they're like, that's why I don't have solar power on this, or that's why I don't have. So if it's worth putting water on grapes, they'll figure out a way to do it. Trust me. Yeah.
Well, thank you very much to all the speakers. That was really great.